Welcome to this particular lecture on Introduction to Industrial Microbiology. In this particular lecture, I shall be discussing the different aspects of industrial microbiology. To start with, I shall be telling you the history and development of industrial microbiology and its subsequent development. Then I shall be telling the different era of industrial microbiology, and I shall follow it up by saying the different arena or different applications of industrial microbiology from which mankind is immensely benefited. To start with, the history or the development of industrial microbiology. Actually, the history no one never knows really when this industrial process of microbiology started. Because people started drinking long time back in ancient Egyptian as well as Roman Empire. People used to drink beer, used to drink alcoholic beverage, and as you can see in this particular ancient wall painting in Egypt, you can see people are drinking beer. Similarly, in the historical monument in Greece, people actually used big earthen containers, and these containers used to carry wine, in which the wine was actually preserved for a long period. So this was actual industrial microbiology starting with fermentation. Subsequently, the history of bread also is very old. This typical leavened bread that was actually done by yeast, and this texture was due to the liberation of carbon dioxide. The technique of bread production is also very old, and nobody really don't know that when this particular technique started. Accordingly, people started growing yeast. So the basic fermentation activity of yeast and its development is very important in the production of alcohol or different alcoholic beverage and as well as different bakery product. Accordingly, yeast being the starter culture, it was maintained in different forms. It can be liquid, or it can be made into block, or it can be granulated yeast. And specifically, this type of commercial granulated yeast is very commonly grown in industry and is sold in packets and can be used in domestic purpose. Next, another very important activity is the separation of fiber by microbial enzyme, as this is a typical stem of flax in which this flax stem contains the secondary phloem fibers. These secondary phloem fibers are attached to the main stem with the help of pectin. So it is the microbe which actually produces the pectolytic enzyme, and this particular microbe is actually called the Clostridium bejerrenki, or there are other species of Clostridium which produces pectolytic enzyme, and the process is called rating. And you can see in this particular scanning electron microscope how this particular bacteria, which is rod-shaped structure, is separating the flax fibers from the main stem. And it is followed up by the production of different organic acid. One such acid is the vinegar. And in fact, the production of vinegar is primarily a two-stage fermentation. Stage 1 is production of alcohol from sugar, and stage 2 is production of organic acid using the wood shaving in which the starter culture of this particular Clostridium was added. And this particular end product is the acetic acid from which vinegar is produced. So this is the bacteria which is the Clostridium acetobutyricum producing the acetic acid of commercial importance. Another very important activity is the production of different alcoholic beverage. Presently we are using different steel fermenter, as you can see here. And this is the process of beer production, which include milling, then mushing, then lautering, then boiling, and then whirlpool, and after that the fermentation, and then maturation, and finally the end product. So basically the production of beer is a very age-old technique. Of course, with time it has developed and has been mechanized. The production of yogurt is a very important phenomenon, and it is also very old technique in which the raw milk, it was actually pasteurized so that the vegetative cells of bacteria are removed, and then the starter culture, that is the yeast or any other bacteria which is going to modify or change or ferment the milk to yogurt, is added, and the raw yogurt is produced, and then it was packaged and it was processed, and sometimes there may be different flavors added to this particular yogurt to make it economically viable. So basically these are certain products which people have been using. But now let me be a little more structured, in which I will be showing the different era or different phases of industrial microbiology. The phase 1, I have actually made it up till 1900. We can call it the fermentation era, when different fermented end products were produced. Phase 2 is between 1900 to 1940, which is the era of antibiotic, because the microbes produce the antibiotics. Phase 3 is between 1940 to 1964, which is the production of single cell protein. Phase 4 is between 1964 to 1979, which is the production of different secondary metabolite of economic importance. But from 1979 onwards, it is actually the era of biotechnology. That means the products are produced by biotechnologically modified organisms. So, to start with, the fermentation era, originally people like Liebig, people like Berzelius, they considered fermentation as a chemical process, quite naturally, because they were pure chemists. But it was Charles de la Tour who actually said that it is not purely a chemical process, but actually it is a biological process where fermentation is the most important process. Subsequently, the discovery of Louis Pasteur between the period of 1857 and for next decade, that is 1867, he made a series of experiments. But the key discovery of Louis Pasteur was that the microorganisms are capable of producing different fermented product, which may include the propionibacteria producing propionic acid, E. coli producing the butanediol, or it is also producing the acetic acid. Similarly, yeast can produce ethanol, and similarly the Clostridium can produce the acetate, acetone, propanol and butanol. So what we can see here, that the basic product of these microorganisms is actually their respiration, which is the pyruvic acid, following normal respiratory pathway. But from this particular pyruvic acid, different types of end products are produced due to different types of fermentation activity of these bacteria and unicellular microbes. So naturally, fermentation actually is brought about by biological organisms. But in this particular fermentation vessel, as shown by Pasteur, the swan neck is very important. He wanted to show that the microbes which are present in the air can easily grow and contaminate the media in which the fermentation will be carried out. By simply bending the neck of this particular retort, you can reduce the surface contamination and thereby make the fermentation more stable. And in fact, this particular application was very useful in preventing the contamination of wine mixture or wine sample in France. Coming to the next era, which is the era of antibiotic, and these are the key players: Alexander Fleming, Ernst Chain and Howard Florey. They discovered penicillin from Penicillium notatum, and what is important, that this particular purification of this drug and production is carried out industrially with a particular process called the staged fermentation, or you can call it the batch fermentation or batch fermenter reactor. In this particular antibiotic production, the batch fermentation means that the starter culture are not added at a time. So the fermentation is carried out for a longer period, and ultimately the end product is coming out and it is purified and sold in the market. So production of antibiotic is the era. Coming to the next important era, which is the production of single cell protein. It started with the discovery of a blue-green algae, filamentous blue-green algal filament, which is spirulina. This spirulina actually is very much enriched in protein, and it also contains vitamin. But what is important, this simple rod-shaped bacteria, which is the Methylophilus methylotrophus, it is producing single cell protein for a prolonged period. So this particular single cell protein can be of tremendous importance, particularly in a country which is suffering from malnourishment. And this is the step of SCP production, where we have the fermentation, filtration, drying, purification of SCP, and of course this process is carried out by submerged fermentation and also sometimes by semi-solid fermentation. But rather this product is relatively easy. After SCP, we come to the next era, which is the production of different secondary metabolite. And this secondary metabolite include the amino acids, the vitamins, the enzymes, the mineral utilizing products, and also the nucleosides. One such important is the production of enzyme, and after that particular enzyme is produced, there are techniques developed by which the enzyme can be encapsulated, and we call it the technique for immobilizing the enzyme. Similarly, production of a polysaccharide outside the bacterial cell in the form of dextran can be used in the production of synthetic cellulose. Coming to the next important product, which of course now is under biotechnological era, is the hemopoietin. Hemopoietin is actually a very important protein, and this particular protein is having multiple role, and this hemopoietin is also produced by microbes by transgenesis. And you know, this particular EPO or hemopoietin can be important for cardiac protection, for angiogenesis, for wound healing, for retinal protection, for renal protection, neurodegeneration protection, and of course most important, the development of fetus and their bone. So EPO that way is a very important product, and if this EPO producing gene can be transferred to the microbe, it can be of tremendous importance to human beings. Similarly, a very important technique, that is production of antibodies, which is monoclonal antibody through the hybridoma technique. What is done? The monoclonal antibody is being produced by binding the antibody producing cell with the rat myeloma, and that will produce huge series of antibodies which are monoclonal antibody. Of course, when we consider this biological product, we also consider certain products which are not purely microbiological, but at the same time they can be transferred or they can be produced because of the successful transfer of the gene. And one such product is the cyclosporine, which is basically a immunosuppressor drug, very important in organ transplant, and the other one is lovastatin, which is reducing the cholesterol level. So these are very important drugs which are also derivatives of industrial microbiology in the present day era. I shall be discussing the different aspects of industrial microbiology, considering the major areas, which may be medical, which may be food, which may be agriculture, which may be industrial, which may be energy generation, and which may also include some applications in criminology. So to start with, the medicinal importance. I have already talked about the production of antibiotics, but sometimes microbes may become resistant to antibiotic. So thereby we started using the different secondary metabolite like the bacteriocin. And this bacteriocin can be of three different classes. The class 1, which is nisin, and this nisin is actually blocking the peptidoglycan byproduct or the peptidoglycan transfer, and thereby the bacterial cell wall is inhibited. Similarly, there may be the class 2, which is the sarcosine, which is causing the membrane depolarization, and there may be the class 3, which is the lysostaphin, which is also causing membrane degradation. So where the antibiotics are not working, these bacteriocins can be of tremendous importance, which are of course generated from bacteria. Another very important application is the production of different secondary metabolite like the insulin production. The insulin producing gene from human or from higher animals can be transferred to E. coli through successful gene transfer, and this bacteria can now produce the insulin. So the price of insulin goes down. So this is a very important application in medical microbiology. Of course, it may also include production of different other hormone, production of vaccine, production of protein, and the list may become endless. Coming to the production of food, I have already talked about the production of yogurt, but at the same time there may be production of different flavored pickle like the sauerkraut. Sauerkraut is actually a fermented cabbage which is produced by microbial activity. At the same time, there may be production of ascorbic acid, And this ascorbic acid is due to the fermentation of glucose to sorbitol, then to sorbose, and finally to gluconic acid, and then to ascorbic acid. And this vitamin C actually is also used as a preservative. It may have medical application and it may also be industrially important. But mind you, this fermentation is carried out by a bacteria called Glucanobacter oxidans. Of course, when you talk about food, the most important food being the vegetable protein, which is the mushroom. The production of button mushroom, the production of oyster mushroom globally is a big industry, which is not only for the purpose of producing protein, but at the also same time it is producing antioxidant and is a very good source of diet or energy to the ailing patients. Coming to the agricultural applications, agriculture actually has two major applications. One is the production of biofertilizer. And these biofertilizers can be of different types. It can include the nitrogen fixer in the form of bacteria like Rhizobium azotobacter. It can be cyanobacteria like Anabaena nostoc, or it can be azolla, which is endophytic, which is also having the blue-green algae. At the same time, it can be phosphate solubilizers, it can be vesicular arbuscular mycorrhiza, which can be plant growth promoting rhizobacteria, it can be sulfur solubilizing, and the list is really endless. So biofertilizers basically help in increasing the soil fertility without causing any chemical damage to the soil. At the same time, we can actually produce transgenic plant having resistant gene from bacteria, and thereby this particular resistant gene will make them naturally resistant against insect pathogen. We can also apply different microbes like this one, the baculovirus, which is a typically budded or occluded virus, and it will produce an occlusion body which is actually killing different insect pests. So basically this is a typical example of a biopesticide. And there may be other forms of biopesticide which is also very important, like the Bacillus thuringiensis, like the Beauveria fungus, which is preventing or killing the Lepidoptera insects. Coming to the industrial product, it includes the different enzymes. Most important enzyme being the cellulose degrading enzymes, which can be exoglucanase or beta-glucosidase, which can be endoglucanase or endo-beta-glucanase, and it can also be the cellobiohydrolase. What is the end product? The end product ultimately will produce cellobiose, and ultimately you can get glucose. So basically cellulose, which is not commercially being used to a greater extent, can generate sugar of tremendous commercial importance, because this sugar can be the source of different fermented end product including alcohol. It can also produce, the bacteria can produce different carbohydrates like the xanthan, which is used in salad dressing, and it can also be a source of potential polysaccharide. The stages of alcohol fermentation, which is a very important industrial product. Basically, it is conversion of glucose to pyruvic acid to acetaldehyde, and finally to ethanol. This entire fermentation process is not only carried out by yeast, but also by different types of bacteria and molds. A very important product coming under biotechnology is the production of golden rice. Now basically the production of golden rice is actually the production of beta carotene. This beta carotene is a precursor of vitamin A. It is starting from the geranyl pyrophosphate, from which the thioin 2 pyrophosphate is produced, which is producing the zeta carotene, finally getting into lycopene, which is also a very important antioxidant, and finally it will produce the beta and alpha carotene. So this beta carotene producing gene, when it is transferred from a particular yeast or daffodil into rice, and expression will produce this golden colored rice, which is actually preventing night blindness in young children in a developing country like India. Production of biofuel. This biofuel basically is the conversion of different Krebs cycle organic acid to different products like the alkanes, like ethanol, like hydrogen, and thereby we can say that this particular microbes can generate a huge number of biological fuel which can be in future day use. And of course, when these microbes are grown in consortia, they can actually complement each other. So basically a single group of organism will produce less number of product, but when it is in a consortia, the end product of one microbe can be actually the substrate of another. So thereby the number of biofuel may increase. Coming to a very important aspect, which is the criminological aspect or identification of an organism by the help of a bacteria called Thermophilus aquaticus. This bacteria produces an enzyme which actually is called Taq polymerase, and it is resistant to heat. It can withstand temperature up to 95 degrees centigrade. Based on this discovery, the technology of polymerase chain reaction was discovered, which is having three major steps, that is the denaturation, the annealing and the extension. But what is the end product? Ultimately, we can produce a huge collection of DNA of the same clone. And this particular technique was involved in the DNA fingerprinting. Coming to the last major aspect, which is the bioterrorism. Now actually bioterrorism is a misnomer. It is the use of biological organism in different activities. One such activity is causing widespread disease. So this particular bacteria of Bacillus anthracis, which is a dreadful bacteria because it causes the disease of anthrax. This can be made into powdered, and this powdered Bacillus anthracis can actually enter into any particular nasal tract and it will cause the dreadful disease and it can be fatal. So in this particular lecture, what we have learnt is that industrial microbiology is encompassing a huge arena. It encompasses food, it encompasses medical products, agriculture, industrial and energy, and even it is having applications in bioterrorism or in criminology. And really speaking, nobody knows when it started, because it started from time immemorial when people first learned the technique of fermentation. So with this particular word, we can understand the importance of industrial microbiology and its huge amount of usage in the present day human society. Thank you.